Hello, my name is Kaveen Suresh, and I'll be going over the light reactions that occur in photosynthesis. This video will cover both linear electron flow and cyclic electron flow. So first, let us discuss linear electron flow. Linear electron flow starts with a photon of light striking a pigment molecule in a light harvesting complex, which you'll see here. This boosts one of its electrons to a higher energy level. And as said, electron falls back to its ground state. An electron in a nearby pigment molecule is simultaneously raised to an excited state. So as you could see, in orange arrow pointed here, meaning the energy was transferred here, creating an excited electron here. And it keeps going. This happens here again. Then as you can see, the arrow points here because it's happening here again. It'll just keep happening until it reaches here, which we will see in the animation. So this keeps happening until it reaches the P680 pair of chlorophyll A molecules in the photosystem 2 reaction center complex. Here it excites the electron pair of this pair of chlorophylls to a high energy state, and the aforementioned electron is then transferred from the excited P680 to the primary electron acceptor, which you can see here. Since the P680 is now missing an electron, it shall be referred to as P680 plus. Okay, so now we'll talk about this molecule of water. So now an enzyme catalyzes the splitting of a water molecule, which enters the photosystem 2. It splits into, okay, so then it'll split. It splits into 2 electrons, and then it'll also split into 2 hydrogen ions and an oxygen ion, which is represented by this red circle. The lettering's a bit small, but it does say that. 2 hydrogen ions and an oxygen atom. The said electrons are supplied one by one to the P680 plus pair, which you will see in this animation. So this keeps happening with each electron replacing one transfer to the primary electron acceptor. So it's important to mention that P680 plus, so P680, as I mentioned earlier, P680 plus is P680 without with one electron removed. So this electron hole has to be filled. It's the strongest, P680 plus is the strongest biological oxidizing agent known. So this characteristic aids greatly in the effort to transfer the electrons from the split water molecule. It isn't included in the animation, but it is important to mention that the oxygen atom that's split from the water molecule immediately combines with another oxygen atom that is split from the same process, so they both form O2. Okay, so each of the photoexcited electrons pass from the primary electron acceptor of photosystem 2 to photosystem 1 via an electron transport chain, which you can see right here. This chain is made up of electron carrier plastoquinone, a cytochrome complex, and plastocyanin. This exergonic fall to a lower energy level will actually provide the energy for the synthesis of ATP, which you will see soon. Okay, so here it is. And then as the electrons pass through the cytochrome complex, the pumping of protons builds a proton gradient that is subsequently used in chemiosmosis. So meanwhile, another photon of light excites a pigment molecule in photosystem 1, and a similar process occurs to when the photon entered photosystem 2. The excitement of electrons once again keeps getting transferred until it reaches the P700 pair of chlorophyll A molecules. And as you can see, just to highlight that the electron is already there, it just becomes excited. I left the electron here. So the electron becomes excited and goes to the primary acceptor. And this creates a hole in the P700 and making it P700 plus. Now P700 plus can act as an electron acceptor, accepting an electron that reaches the bottom of the electron transport chain from photosystem 2. So the electron transport chain we just saw, where the electrons got transferred. So now the photoexcited electrons are passed in a series of redox reactions for the primary electron acceptor of photosystem 1 down to a second electron transport chain through ferredoxin. It should be noted that this chain does not create a proton gradient, so it doesn't produce ATP like the electron transport chain we just saw. So as you can see, it passes through, and then it passes through ferredoxin, and then it'll reach the NADP plus reductase. So now the enzyme NADP plus reductase catalyzes the transfer of electrons from ferredoxin to NADP plus. So this will create NADPH. So as you see, what happens is the 2 electrons are necessary for its reduction to NADPH. This molecule is at a high energy level, so NADPH is at a high energy level than water, and its electrons are more readily available for the reactions of the Calvin cycle than were those of water. Okay, so that was the entirety of linear electron flow, and it created ATP and NADPH, which will be used in the Calvin cycle, which I'm going to make another video for soon. Okay, so now let's discuss cyclic electron flow, which is similar to linear electron flow, but it does have a few differences. So first I'll have a diagram. Okay, so in certain cases, photoexcited electrons can take this alternative path, so cyclic electron flow, which uses photosystem 1 but does not include photosystem 2. So as you can see, photosystem 2 is crossed out, and photosystem 1 is just, I drew out a cycle here. Several existing groups of photosynthetic bacteria are known to have only photosystem 1. For these species, cyclic electron flow is the sole mean of generating ATP. So it is very important. So the process is going to be a little simpler to explain, but photoexcited electrons from PS1 are occasionally shunted back from ferredoxin to chlorophyll via the cytochrome complex and plastocyanin. This electron shunt supplements the supply of ATP via chemiosmosis, but produces no NADPH. So as you can see, it created ATP but no NADPH, and process is relatively similar. And you see the excited electrons get passed on, like in the other one, and then the cycle begins again with the electron there. Okay, thank you, and that was my video on the light reactions of photosynthesis. Next, please do watch my video on the Calvin cycle. All right, thank you.